undergraduate and PhD in physiology from the University of Leeds in the United Kingdom, and then moved to do a postdoc and beyond, I think in Maryland with John Lederer. And John Lederer, I think that was the man that time there, calcium. So there was a lot of work on calcium and IK-ATP also. And I have to say that Colin has done beautiful work on ion channels, potassium channels in the heart, on inward rectifiers, and in particular IK-ATP. And I ran into his work when Robin Shaw, one of my PhD students, and I simulated the effect of ischemia, acute ischemia, on the action potential of cardiac myocytes. And of course, we based it on Colin's data that even then showed some miracles that IK-ATP can perform, such as shortening APD by 50% by opening 1% of channels. So anyway, without further ado, I would like to give the plugin that is missing to Colin.
Thank you, Yoram. Well, I'm very honored to be the first one for this session. I guess I'll put the glass down. And in the spirit of this thing, what I'd like to do is, since we're all in-house, I would like to actually show some ideas and hopefully trigger some discussion. Closed everything down. I gotta find where I am now. I would like to hopefully initiate some discussion on the question that Richard brought up, which is, what does this channel do? The short answer is, we don't really know, although we've spent an awful lot of time trying to find out what it does in the heart. And it's still very surprising, and we still don't really know what it does. And so I'll go through the data. So I don't think we need this slide. This is just a standard slide that I show to people to try and introduce different concepts of functional variety of potassium channels. There's several people in Yoram's lab have worked on modeling delayed rectifier or voltage-gated potassium channels which are involved in action potential repolarization. This would be a model of the cardiac action potential. These are potassium channels which are closed at negative resting membrane potentials, open when the cell depolarizes, and that's why they're involved in repolarizing. The KATP channel is part of a group of channels which are termed inward rectifiers by virtue of the fact that they tend to show inward currents that are larger than the expected outward currents if we had a normal GHK rectification of the channel. So these channels are open at negative voltages, tend to close down at positive voltages, and broadly fall into two functional subgroups, those that are strongly inwardly rectifying, which includes the classic IK1 in the heart and IKACH. These are channels which are involved in stabilizing the resting potential that close down very strongly at positive voltages so they don't impede the long depolarized phase of the action potential. The KATP channel shows only very weak inward rectification, and physiologically over the relevant voltage range from, say, minus 80 millivolts around the resting potential up to plus 50 millivolts at the peak of the action potential, it's more or less a linear conductance that doesn't significantly rectify. And so it will not only stabilize resting potentials and, if activated enough, actually stop the generation of an action potential, but it will, as Yoram pointed out, cause significant shortening of the action potential as well. What's the point of this? In the heart, major K conductance at rest results from strong inward rectifier or from weak inward rectifiers. Okay, this is just to say that the resting potential is generated by these channels rather than voltage-gated channels. And to put you all on the same page in terms of the structure of these channels, because I'm going to talk a little bit about structure function, starting from the biggest members of the cation channel superfamily, the sodium channels and the calcium channels, which by a quirk of the history of all this, they were the first ones to be cloned. They have the whole channel is encoded in a single polypeptide which contains 4 pseudo-homologous domains, 1, 2, 3, 4 four of these four all come together to generate the channel, and in the potassium channels, the first clone potassium channels with a voltage-gated shaker channel subfamily, and there we see a single one of these domains in a single polypeptide, and we have four of these subunits then come together to make the channel. There are 6 transmembrane helices. The amino terminus and the carboxy terminus are all intracellular. There is a large loop between the 5th and 6th transmembrane helix, which generates pore lining residues. And in the last channels to be cloned, which are the simplest structurally, the inward rectifier channels are all encoded by subunits which have this transmembrane topology, 2 transmembrane helices, internal amino and carboxy termini. These 2 helices correspond to S5 and S6 in the voltage-gated channels, and that extracellular loop, although in this picture, which came from an old textbook, they didn't realize this, we now know that, of course, this loop dips back into the membrane, and that's what's generating the selectivity filter of the pore. 2 years ago, Declan Doyle and colleagues crystallized the first member of this KIR channel family. They crystallized the bacterial homologue, and we are now using this as a model for all of the eukaryotic channels. What you can see in this ribbon diagram in 4 different colors, in yellow, green, pink, and blue, you see the transmembrane helices. So this would be the membrane here. This H5 loop is what is dipping back into the membrane here to generate this very narrow selectivity filter, which is where potassiums are selected against other ions. Then there is the cytoplasmic domain is made up of the amino and carboxy termini together, and it looks sort of like a rat's nest on this picture. You can't see much of it. It's a whole series of beta strands. This is more of an advert than a result. In our own lab, we were also chasing the crystallization, and we cloned the same channel. We expressed it as a His tag with a thrombin site protein in bacteria, purified it. We have a very pure preparation. This is a Western stain against the His antibody, and you see a pure protein band on Ponzo S staining. This is the work primarily of Desha, Jaya, who's left us, and Yana, who has also gone back to the clinic. Desha is in the audience somewhere. He's a nephrologist here in the renal division. And Jaya succeeded in crystallizing it. There's a crystal. This is a diffraction pattern. And this is the preliminary electron density map with Declan Doyle's model inside it, just to show that, in fact, we get the same. We have an electron density map that matches what they got. So we've crystallized the same protein. And I'm not sure why I'm showing you this. It's because it was in the seminar, but the one I picked up. But I want to make the point that we are using this model, and with this we're going to be able to build up a model of what's happening in the eukaryotic channels. We were able to get functional data on the channel. Nobody else has published any yet. We incorporated the proteins into liposomes, and we made the liposomes in a high K conducting medium, and then we removed potassium from the outside. This will cause potassium to leave the liposome, so that leaves behind an electronegativity inside the liposome, which provides a driving force for cations outside to move into the liposome. So then we place the liposomes into a solution containing rubidium, 86 rubidium, which is radioactive. The rubidium will go into the liposome if there is a pathway to do so. So if the channels are there, then they will go in. The rate at which rubidium is taken up into the liposomes will be proportional to the conductance in the liposome membrane. And to cut a long story short, we've shown by this method that this KR-back protein can support a robust uptake of rubidium, which is blocked by barium. KCSA is a positive control. This was the first crystallized bacterial K channel. That also supports uptake, which is barium blockable. If we don't have any protein in the liposomes, we get no uptake. If we make the liposomes in potassium or rubidium or cesium, we get uptake, but not if we make them in lithium or sodium. So lithium and sodium are not conducted by the channel. That's the basis of this result. And if we do it the other way around, which is to make the liposomes in potassium, remove the potassium, and then add external cations, which will dissipate the gradient if they can go through the channel. Potassium, rubidium, and cesium will dissipate the gradient, but lithium, sodium, and N-methylglucamine will not. So this just shows that it's a potassium channel. And I was talking to somebody today in Yoram's lab about our latest set of data with this, which is looking at regulation of this channel by PIP2, but that's another story. Now to come back to the KTP channel, so I said that all of these inward rectifiers, the channels are made of members of this KRS subunit family, and KRS 6.1 or 6.2 are the subunits that make up the KTP channel in tissues, and KRS 6.2 is the subunit that makes up the cardiac channel. That's what I spend most of the time talking about. The channels are tetramers of these KRS 6.2 subunits. The KTP channel is unique in that each of these KRS 6.2 subunits is also associated with this large ATP binding cassette family member called the sulfonylurea receptor, and that also comes in two flavors, SUR1 and SUR2. It turns out that KRS 6.1 and SUR2 are immediately adjacent to each other on the chromosome, and KRS 6.2 and SUR1 are immediately adjacent to each other. And I'll come back to this at the end, because I have some ideas, and so I'd like to discuss this, about the possibility that these are actually co-translated. Anyway, the sulfonylurea receptor has these two nucleotide binding folds in the cytoplasmic domains. We have good evidence now, and a lot of it's some of it is coming out of the work of Ricard Massio, who's a graduate student in the lab, that these actually make a functional dimer. Nucleotide binding folds in these ATP binding cassette family of proteins generally are capable of hydrolyzing ATP, and then can be trapped in a post-hydrolytic state by the binding of magnesium ADP. The channel portion is where ATP inhibition resides. So I don't know if I have that on the next slide. Okay, let me just go through this record, which shows you the essence of how the channel is regulated physiologically, at least in our experimental conditions. So this shows an inside-out patch clamp recording from a cell. A membrane is excised from a cell, COS cell, which is a monkey kidney cell, expressing these KTP channels at a very high density. In this case, we're expressing SUR1 and KRS 6.2. We have half a nanoamp of current, which is corresponding to about 150 channels in the patch. So you see the more or less macroscopic current. You don't really resolve individual channel events. When we apply 1 millimolar ATP, the channels are almost completely inhibited. 100 micromolar ATP, there is some activation. But then in the maintained presence of just 100 micromolar ATP, if we now add 500, this should be micromolar magnesium ADP, not millimolar. We add a small amount of magnesium ADP, and now we get this strong activation of the channels, and everything is reversible when we put the solutions back again. So this shows the essence of how we think the channels are physiologically regulated. They are inhibited by ATP binding to get is that the half maximal concentration of ATP to inhibit the channel is typically around 10 micromolar, and magnesium ADP shifts this value to the right. So in the presence here of 100 micromolar, we've got substantial inhibition, but with magnesium ADP present, we almost completely activate the channels. Okay, so the main purpose was to talk about KTP channels in physiology and to talk about their role in the heart. KTP channels are present in most tissues. They're present in the heart, skeletal muscle. The KRS 6.2 SUR2 combination is involved in the response to ischemia and perhaps in exercise adaptation, and maybe Yoram has some opinion about this, but these channels, I should say, and you will agree with this, they're present in the heart at an extremely high density, higher density than any other K conductance, and at such a density that if you activated enough of them, you can completely stop the action potential. And teleologically, it doesn't make sense that they're not there for no reason. Too many negatives. That they are, teleologically, it only makes sense that they're there for some reason. And that's the question we're going to search. In the vascular system, they're involved in ischemic and perhaps humorally stimulated vasodilation. Absence of these channels may actually cause prince metal angina, or they cause a symptom like that in mice. And it's possible that they are underlying something similar in humans. In the central nervous system, again, they're involved in the response to ischemia, response to CNS glucose levels, and in the pancreas, which I'll talk about first, they're involved in the response to blood glucose levels in alpha and beta cells. I guess I didn't stress the point that because they're regulated by nucleotide levels, they're inhibited by ATP and activated by ADP. This means that they're going to respond to the cell's metabolic state. So I haven't detailed the question, but the question that we sought to address is what is the role of these channels in physiology of tissues, and what happens then if we alter the physiologic regulation of the channels. And the driving force behind a lot of this was, first of all, to prove what I'm going to tell you is the paradigm for how KTP channels are involved in glucose sensing in the pancreas, but secondly, to address the question of what are they really doing in the heart, where we see this massive density, but under normal conditions, as Joran said, they're not open, and so we don't see what they do. So our goal was to tweak the system by making them less sensitive to ATP so that they would become active. And in the course of a lot of mutagenesis that we did early on, those pictures. Oh, there it is. Bo Costa, who doesn't look like this anymore, he's sitting over there somewhere, did an awful lot of structure-function experiments. He does actually, he hasn't aged. He did a lot of structure-function studies on the KTP channel when we first began working on the clones. And amongst the mutations that he looked at, he identified two which had interesting effects on ATP sensitivity. A deletion of an internal 30 amino acids from the Qrs6.2 subunit caused about a 10-fold shift in the ATP sensitivity of the channel. So this is a dose-response curve to ATP, and as I said at the beginning, you get about half maximal inhibition at 10 micromolar. If you delete the N-tonus, it shifts tenfold to about 100 micromolar in our recombinant expression. If you combine that with a mutation of K185Q, which we now have good reason to think is actually in the ATP binding site itself of the channel, this double mutation shifts ATP sensitivity more than 100-fold up into the millimolar range. So Bo took these two mutations and tagged them with GFP at the C-terminus and made what I think was the first GFP-tagged transgenic animal, actually. This just shows that the GFP tag doesn't alter the recombinant behavior of this deltran 30 construct. He took the deltran 30 or the K185Q mutation in combination and put them behind the control of the rat insulin promoter or the alpha myosin heavy chain to drive expression respectively in pancreatic beta cells or in the heart. And just to fill you in on what was the paradigmatic understanding of KTP channels in control of insulin secretion. When blood glucose goes up, it's taken up by beta cells, it's metabolized, this causes a rise of ATP, a fall of ADP, and what I just told you about the way that the channels are regulated, the rise of ATP will tend to close the channels, and the fall of ADP will tend to reduce their opening, so the net effect, of course, is closure of the channels. Closure of the channels will cause depolarization of the membrane, activation of voltage-gated calcium channels, a rise of intracellular calcium, and that's the trigger to insulin secretion and compensatory lowering of blood glucose. So we asked the question, well, if we made the channels insensitive to this closing effect of ATP, would we get a reduced insulin secretion? Here's the prediction, that we'd get an increase of glucose, would cause a change of ATP and ADP. If this failed to close the KTP channel, the cells would then remain hyperpolarized. We'd block calcium entry, block the rise of calcium, block the release of insulin, and fail to lower glucose. We might even get a diabetic phenotype. And in fact, we did get an extremely profound diabetic phenotype. So, to cut a long story short, when we use the double mutation, the deltran 30 K185Q mutation, which shifts ATP sensitivity about 100-fold, we got no founder mice from 60 live births from over 200 embryo injections. So we gave up. When we looked at the single deltran 30 mutation that shifts ATP sensitivity tenfold, we got 5 founders, and 4 of them were viable, probably mosaic, for reasons that I can come back to, but every one of them gave offspring that died within 4 to 7 days. So we had 2 males, 2 females, we got lots and lots of pups, but every one of them died within a week. So in a bit of a heroic effort, Bo started looking at these animals at day 2 when they're about nothing. And what he found was that measuring blood glucose, although in control, this shows individual measurements, this is the average. Control blood glucose is about 120 milligrams per deciliter, which is I think about 6 millimolar, and normal for glucose. If you looked at the transgenics, almost every one of them was above the level of detection, above 20 millimolar. So they had extremely high blood glucose. When he measured insulin levels, here again is individual measurements, they're very variable, but averaged around 500 picograms per mil, which is normal. And the transgenics were at or below the level of detection on average. And if you measured blood ketone levels, they were very low in control, as they should be in a normal animal, but they were very high in the transgenic, which means that they're metabolizing fats. So these are the symptoms of an extremely diabetic animal. Very high blood glucose, no insulin. This is like a profound type 1 diabetes. When Bo took pancreatic samples and made slides with histology on them, staining for insulin, here's a control slide showing two islets, and you see this brown staining for insulin. He did the same thing with the transgenics. There's an islet, and it stains just as well for insulin. So the islets had insulin in them. And here's a double staining for insulin and glucagon from one of these transgenics, where there is good insulin staining in the middle of the islet, and then there is glucagon staining around the periphery. This is the normal morphology of islets, so the morphology is not altered. So we had a very clear picture. Very high glucose, no insulin in the blood, normal insulin in the pancreas. So these animals are profoundly diabetic, but they've got insulin. Here's one. We did actually have 3 transgenic animals that made it to weaning. This one died a couple of days later. It's about half the size of its transgenic glutamate, probably because it doesn't have any insulin to support growth. And in a final tour de force, Bo actually isolated some beta cells from these 2-day-old mice, and actually was able to patch them, and he found a couple that were green, and when he did, he measured the ATP sensitivity of these of the KTP channels in the cell membrane. Here's a control membrane. You see very high sensitivity to ATP, 10 micromolar here gives just a little bit of activity. In the transgenic, here he sees almost maximal activity at 10 micromolar and half maximal activity at 100 micromolar, which is what I said was the in vitro ATP sensitivity of this mutant. When we averaged all the data together, what we saw was 10 micromolar is about the half maximal inhibition for the wild type channels, but in the transgenic membranes, there was a very slight shift to the right, about 5-fold on average in the ATP sensitivity. So these results had very profound implications. It said that the cells are actually normally sitting on a knife edge, that if you make cells slightly insensitive to ATP, what you get is failure to secrete insulin. This suggested this could be a cause of a human disease, and we predicted this, and we've been saying it actually for several years in seminars. I've been saying it in seminars, of course, but we've also said it a couple of times in reviews, but the field, the clinical field, had failed to demonstrate anything. But just last year, it transpired, it was fine. Andrew Hattersley in England finally got hold of neonatal diabetics and started typing them, and in fact, these mutations, gain-of-function mutations of KTP, cause transient and permanent neonatal diabetes. This is a disease, this is a rare genetic disease, but it's just the same as the mice, characterized by no insulin secretion in parallel with low glucose. It's detected normally between birth and a year old. It can be permanent or transient. These babies have very high blood glucose. They often have ketoacidosis. The treatment objective has always been to maintain normal blood sugar, so these children have always been given insulin injections, which is to a clinician the obvious natural thing to do. They think it's like an early onset type 1 disease. But now it's all changing, of course, because now we know the disease is caused by KTP channel mutations. And KTP channels, I told you they're made of the sulfonylurea receptor in the channel, and the sulfonylureas are very potent blockers of this channel. So now these babies are being weaned from insulin onto sulfonylureas, and so now they take sulfonylureas in their milk and they don't have to be injected 3 times a day with insulin. So it's very nice that this has led to this kind of realization. That's the first paper in the New England Journal. And it turns out that these mutations are dotted around throughout the KTP channel. Here's where the ATP binding site is, and I said I didn't say it, but there is 1 per subunit, so there are 4 on the channel. And this is a linker region, which I forget who it was I was talking to today, but that paper that's just come out. I'm digressing. Let me just say that these mutations are dotted around the channel. Interestingly, 3 out of the time I made this slide, 3 out of the 8 reported mutations were all in the ATP binding site. So a lot of them, all of these mutations cause loss of ATP sensitivity, and many of them cause it by actually impeding the ATP binding. The results have several implications. First of all, this could be the tip of the iceberg. So the first reported mutation caused quite a large shift in ATP sensitivity. Our mice suggested that even a 5-fold shift in ATP sensitivity would be enough to cause the disease. We could ask the question, well, what happens at lower levels of KTP overactivity? If we made it only 3-fold or 2-fold or 1.5-fold overactive, is this going to impair insulin secretion? Could this be involved in, say, type 2 diabetes? Secondly, as I've already alluded to, these diseases can now be treated by sulfonylureas, not insulin. And thirdly, of course, excitability can be modulated by many factors. So, although it's now shown that that gain of function of KTP causes this disease in humans, it could equally happen by loss of, say, calcium current activity, because of course it's the interaction of calcium channels and K channels that are regulating excitability and hence regulating insulin secretion. So what happens at lower levels of KTP activity? What I said at the beginning of this part was that we got 4 founder lines that gave the same phenotype. We had a 5th one, which we thought was perfectly normal, but we've since done glucose tolerance tests on it and shown that it's actually mildly glucose intolerant. We've now also generated 2 new lines, F line and G line, which also appear to be normal, but Bo did sort of a case study on one transgenic family and found following 3 transgenics and 3 wild-type littermates in a family after weaning for about a year, he saw that the wild-type littermates, which are in black, never have blood glucose above about 180 mg per deciliter. That's normal. But the F-line mice would occasionally show these extremely high diversions, and they could have blood glucose up above 500 mg per deciliter, which is 30 millimolar. This shows that these mice are actually glucose intolerant, and when he looked at them on average, the wild type, when you do a glucose tolerance test on them, you inject them with glucose, blood glucose goes up, and then comes down very gradually. In the transgenic mice, it goes up higher and comes down more slowly. So, and what I should say is that in these lines of mice, when we look at the level of expression, have now shown that there is a human polymorphism, E23K, which actually happens to be in the N-terminal 30 amino acids, which shows an association with type 2 diabetes. And if you're homozygous for E23K, and about 13%, I think, of Caucasians, which is, so it's probably half a dozen people in this room, will be homozygous for E23K, you have a double the chance of getting type 2 diabetes. If you're heterozygous, you have something like a 1.5-fold chance of getting type 2 diabetes over the E23 normal variant. And this is Bo's data. It's also been published by Schwanstecker that the E23K mutation actually shifts the K1/2 from about 10 micromolar to 17.6 micromolar. So it shifts it about almost 2-fold. So just a very slight shift of ATP sensitivity in the right direction to make cells less likely to secrete insulin, and this is associated with type 2 diabetes. So, general idea is that KTP overactivity could underlie a whole spectrum of diabetes, from normal, and then the E23K variant might predispose to type 2 diabetes. These mutations, as Bo has now looked at, are the less severe, tend to be less severe shifts in ATP sensitivity. These underlie transient neonatal diabetes. The permanent neonatal diabetes has the more severe shifts, and the most severe shifts cause this syndromic version of the disease, which includes neurologic and muscular features. And I will say that the muscular features, at least the muscular weakness, has been treated by giving these patients sulfonylureas, probably because it's blocking the KTP channels in other tissues, whereas insulin never treated that form of the disease, never treated those symptoms of the disease. Now we come to the tragic thing. As Yoram said at the beginning, and this is the paper he was referring to, reduced ATP sensitivity in the heart. Oh, sorry, that's not what he said. What he said was that reduction of activation of 1% of the KTP channels, the total KTP conductances available, can shorten the action potential duration by 50%. And we have shown this experimentally, and what you see here is recordings from a rat ventricular myocyte exposed to metabolic inhibition at different times. The action potential is getting shorter and shorter. This is because KTP channels are being activated. And here is my early modeling efforts, which have been done much more sophisticated ways by Yoram and colleagues now, to include a KTP conductance, a linear conductance, into this case Dennis Noble's Fox-Soft heart model, and then scale the amplitude of the conductance by the ATP sensitivity of the channel under pseudo-physiologic conditions. And the essence of the point is that at normal ATP concentrations in the middle 5 to 10 millimolar range, we expect to have almost no channel activation, and we'd have a normal action potential duration. But even within the millimolar range, if the action potential drops, and if ADP rises, we will tend to expect to see action potential shortening, and we would expect to see about 50% action potential duration of 1 millimolar ATP. So, given that, if we took those two transgenes that I told you about at the beginning, deltran 30, which shifts the ATP sensitivity tenfold, and deltran 30 K185Q, which shifts it 100-fold, so that we now have half maximal activation at 4 millimolar, we should have very significant activation of the channels under physiologic conditions. And our prediction was that it would be deadly and we'd never get any transgenic animals. And it was wrong. So in fact, we found that transgenic hearts expressing the double mutation seem to be almost perfectly normal. Now these are telemetric recordings of electrocardiograms from wild-type hearts, and in this case, line 2 transgenic hearts. We had 4 separate lines, and they're all more or less the same. These were recordings done by Catherine Yamada, and the essence of what we found was that there was a slight, about 15% reduction. These are littermate comparisons from line 2 or line 4 mice, 2 different founder lines. Whenever we compared littermates, we had about a 15% reduction of the overall heart rate in these conscious recordings. And this is the average data, where the transgenics were slightly slower. If we looked at isolated myocytes, we saw a very high fluorescence of the GFP. So we have a very high level of expression of the transgenic protein. If there's any mitochondriacs in the audience, this shows mitotracker red, which I think is basically rhodamine, staining mitochondria in these long strands in the cell, and there's no overlap between the mitochondrial staining and the GFP staining, so we don't think these are in the mitochondria. We know that at least some of them are in the surface membrane, because we do in fact get a big change in the properties. And this is the most striking, well, one of the very striking pieces of information in this talk. We get very big changes of ATP sensitivity. Here's a recording from a wild-type isolated myocyte, a bit like the one I showed you at the beginning, looking at the ATP sensitivity. Patches excised at the arrow, channels activate, at 5 millimolar ATP, they're completely inhibited. They're a little more than half maximally inhibited at 50 micromolar. On average, in the heart, the half maximal inhibition is around 30 micromolar, a little less sensitive than the recombinant channels in COS cells, but similar, around 30 micromolar. Here is an excised membrane patch from one of the line 4 myocytes. This is one of the high expressors. There's no real channel activity on cell. Patch is excised with the arrow. Now though, 5 millimolar ATP doesn't completely inhibit the channels. 50 micromolar, which gave more than half maximal inhibition here, doesn't inhibit at all. 500 micromolar is very little inhibition, and on average, the line 4 is around 2 or 3 millimolar half maximal inhibition. So we have got about, on average, about a 60-fold loss of ATP sensitivity, which is huge, and which all of our modeling efforts would have argued should have caused significant action potential shortening. There is one twist to this, which is that as the ATP sensitivity is reduced in these different lines, the white shows wild type patches, the green, or the gray shows intermediate expressors, and the black shows the highest expressing lines. There is a negative correlation between the K1/2 and the current density. What this says is that there is also some dominant negative effect of these transgenes, which is that as more and more of them get incorporated into the surface and make the channels less sensitive to ATP, they reduce the overall density. And now, this is the work that was again, those transgenes were made by Bo, and now Tom has done most of the next set of work, which is to characterize in more detail what's happening in these hearts. Tom measured action potentials, both surface action potentials on intact heart in collaboration with Anatoly Lopatin, who's now up in Michigan, and he also measured with whole cell clamp the isolated cell action potential duration. The main point was that in these transgenic animals, there is no action potential shortening, whether you look at isolated cells or if you look at surface ECGs in the heart. So even though we have very ATP insensitive channels, you don't get any action potential shortening. When Tom looked at KV channels, he looked at transient output currents, comparing wild type to line 4. What he noticed was that although the time-dependent currents don't look obviously different between the two, there tended to be a slightly bigger steady-state current in the transgenic myocytes compared to the wild type, and averaging the steady-state currents from multiple cells from several animals of both wild type and line 4, the difference currents showed a slightly flatter wild type compared to the transgenics. And if you look at the difference between subtracting the wild type from the transgenic, what you see is a current that rather resembles what we expect the KTP current to be. That is, it's going to reverse somewhere down here negative. It appears to reverse at minus 50, but it's difficult to get it correct. This might be somewhat asymptotic, so I wouldn't make too much of minus 50. The point is it reverses at a rather negative potential, and then it shows a very weak inward rectification at positive voltages. So this looks like what we've got is a very low level baseline activation of KTP. This half a nanoamp is very small compared, it's a big current, but it's a very small current compared to the overall availability of KTP, which could be as high as 50 nanoamps or more. So it's less than 1% probably activation of total KTP conductance. But it is a slight increase in background currents. The plot thickened when Tom looked at calcium currents, and he looked at contraction in these myocytes. And the main, actually preceding this with Polly Hoffman in Memphis, we looked at contractility of the whole heart, and what we showed was the contractility of the transgenics is actually increased. Tom showed it again at the single channel level, and one of the points he noted was that with wild type channels, this is now recording of single myocyte contractility with video microscopy. If you put isoproterenol on, you get a stimulation of contraction in the wild type. There was no stimulation of contraction in the transgenics. Stimulation on average in the wild type about 50%, no significant stimulation in the transgenics. Then he looked at L-type calcium current. Isoproterenol gives about a 50% stimulation of the wild type current, but it gives no stimulation of the transgenic current, and the transgenic currents on average are much bigger in the first place than the wild type channels. So the calcium current is somehow pre-stimulated in these myocytes. So what we think is happening, and what I think this leads us into, is complex remodeling of what happens when we manipulate these K conductances. There have been there's been a lot of knockout animals and dominant negative animals made to manipulate K conductances in the heart, but there's been relatively few examples of K channel overactivity. And only this, and as I'll come back to it at the end, an IK1 overactivity in were generated by Anatoly Lopatin have really looked at increases in any background conductors, and they get a similar phenotype. So anyway, we think that what's happening is we've got a very weak background activation of KTP. We can't explain yet why we don't have massive activation, and there is a compensatory calcium current change. Now, so we've got a strikingly different phenotype, that if we make a little bit of overactivity in the pancreas, we get this neonatal diabetes and death. If we make a large overactivity of KTP, or at least a large loss of ATP sensitivity in the heart, we get a relatively benign phenotype. And one important point that might bear on this is the fact that the KTP channels in the heart are made of different subunits. So, and the pancreas. So in the pancreas, I said it's Qrs6.2 plus SUR1. They're the main, probably even the sole constituents of the KTP. In the heart, it's Qrs6.2 and primarily at least SUR2A, the SUR2 subunit. And one of the things that we noticed, I think Daisha, who's sitting in the back of the room, I don't do any of this, by the way, it's everybody in the audience who's done all this. Daisha first noted that SUR1-based channels are much more effectively activated by ADP. This is a record like the one I showed at the beginning, where we see ATP inhibition, then apply ADP, and we get big activation of the channels. They're much more effective at activating channels than ADP is at activating SUR2-based channels. And work we've done a long time ago argued that, in fact, physiologically, it's not so much a change of ATP that's going to regulate channels, because ATP doesn't really change much in cells. It's the change of ADP that's going to be critical. And so you can see that ADP is not activating as well. And when we average data, this is now the work of Ricard Massier, who's a graduate student, MSTB student in the lab. We see about what looks like about a 10-fold shift to the right in the ability of ADP to activate SUR2 channels and SUR1. So at least part of the difference might be that ADP stimulation is less effective in SUR2-based channels than in SUR1. So, with that in mind, Tom made a new set of transgenic animals where he now overexpressed either SUR1 or SUR2 in the heart under control of the myosin heavy chain promoter. And I beat him to put a his tag on it, but he insisted on putting a flag tag on it. He put a flag tag on it. Anyway, so he generated these lines whereby RNA levels, he got a lot of overexpression of SUR1 in the heart. He could get up to 150-fold over whatever the endogenous expression was by RT-PCR. And he got multiple lines with differing levels of expression by RNA. And at the protein level, you can see that that corresponds to differing levels of protein. This is the work of Maria Sarah-Remedy, who's also in the audience. And I want you to concentrate on two lines, the 720 line, where we have massive overexpression of the protein, and the 697 line, which actually gives very little overexpression by RNA and relatively little protein by anti-flag Western. And I'm going to show what happens with those two lines. The first point to note was that we get, as with Qrs6.2 overexpression, we get a dominant negative effect of these transgenes. The low level 697 gives a very weak and actually non-significant reduction of whole cell KTP conductance, but line 720 with a high level expression of SUR1 gives a much bigger knockdown of whole cell and of single channel conductance. By the way, our goal was to, if you haven't figured it out, was to replace the wild type SUR2 with SUR1, which is the pancreatic isoform, hoping to reiterate the dramatic phenotype that we see of pancreatic channels. So what's causing this dominant negative effect of SUR, as we showed for Qrs6.2, this is a cartoon of Tom's. I'm not sure whether it really makes the point, but the general idea is, and this encompasses a lot of work that's been done by Lily Jan and others, that each of the two subunits, Qrs6.2 and SUR1, contain an RKR sequence in them, which is this cationic sequence, which causes each of those two subunits to be retained in the endoplasmic reticulum when they're expressed on their own. But each of those two retention signals are mutually masked when you get SUR and Qrs6.2 co-expressed, and that mutual masking allows them to export ER and transport to the surface membrane. So what we think is happening in these transgenics, when we overexpress 6.2, is we have too much 6.2, not enough SUR to go around. When we overexpress SUR, we don't have enough 6.2 to go around. So that's why we have dominant negative effect. So what's the obvious next experiment? We cross the two mice. Now we're going to have overexpression of Qrs 6.2 and of SUR. And the idea would be that we should now generate a big overexpression of overactive channels. Well, we didn't get what we thought. And we did get something interesting. So some crosses actually never generated double transgenics. Remember I pointed out the two lines, 697 SUR1 is low level expressing, 720 is high level expressing, and in the Qrs 6.2, line 2 is relatively low and line 4 is high overexpressing. And if we crossed line 4 transgenic animals, this is overexpressing the insensitive Qrs 6.2, with 720, we never got any double transgenic mice. But if we crossed line 2 and 697, we got them appearing at the frequency that we expected. If we crossed the high 6.2 with the low SUR1 or the high SUR1 with the low 6.2, we got less than the expected number. So there was some lethality to mixing the two. And when we looked at this over time, Tom spent weeks going in and out of the animal house to track this. Crossing 697, which is the very low SUR overexpressor with the line 4, we found that we got death at an average of about 8 weeks, and didn't get so many animals, didn't do so many litters to look at this. Crossing the high SUR with the low 6.2, again, we got aggressive death of the animals. And we called it a sudden death because there was no obvious phenotype to the animals when they died. They'd look normal one day, you go into the next day in the cage, and they're dead. And so the question is, what has gone wrong? Have we in fact generated, oops, no, that's preemptive. So have we in fact now generated massive overexpression of KTP and killed the animals in the ventricle? The answer is not in the ventricle at least, because what we see, and I'm not showing you all the data, we get an even stronger dominant negative suppression of ventricular KTP. So I don't have the data to show you, but you have to take my word for it, that when we patch ventricular myocytes, now we see essentially no KTP in the ventricle. But something has gone significantly wrong electrically. And we get quite complex ECG phenotypes in these animals. We've looked at them in a virgin anesthetized animal and in telemetrized animals. This is recordings of ECGs from wild-type animals at different time scales. And if you see a normal-looking rhythm, and if you look at them at a fast time scale, you see the atrial beat, the Qrs complex, and this kind of T wave that you see in mice. This is a common finding in these mice, that they have a 2-to-1 AV conduction block, and here you see on a slow time scale this mouse has a regular T wave. Did I say T wave? No, sorry, a regular P wave, but not every P wave, like this one, this one, this one, this one. Not every P wave is followed by a Qrs complex. In other words, there's a conduction block between the atria and the ventricle, and it's intermittent. So sometimes we'll see two ventricular beats and then a gap, and then 3 and then a gap. And one of the things that we have noticed, though, is that is tolbutamide sensitive, and this is one of the potentially interesting points, which suggests that it's actually KTP channel activity that's doing it. So in the wild type here in control, this is a regular recording, and then we apply tolbutamide to the wild type animal, and what we get is a speeding up of the heart rate. It drops from about 160 milliseconds to 120 milliseconds beat to beat interval. Here's one of the transgenics, which has got 2 second degree and occasional third degree block. So if you look at the RR interval, that's the interval between one of the Qrs complexes and the next. Most of the time it's around 180, which is normal, tracking the atrial rate. Occasionally it jumps, and this is time here, so each one of these is one RR interval. Sometimes it jumps to double that duration. That's when it's skipped 1 beat. Occasionally it'll jump to triple that duration. That's when it's skipped 2 atrial beats. And here we applied, the animal was given tolbutamide just before think, at the beginning of this record, and you can see that at this point it's got 1, 2, and occasional 1 and occasional 2 missed beats. But then within a minute or so, the timescale's missing off here, I'm afraid. This is about, what was this, Tom? About 3 minutes, probably? 5 minutes? The timescale's missing, sorry. But anyway, within a minute or two, what you see is that these missed beats disappear and it drops to a regular rhythm. Now, we can talk about this in detail, and we have to do more experiments, because this speeding up that you see even in wild-type animals of the heart rate could in itself cause some loss of these skip beats, but it is suggestive that what's happened is we've got some excess KTP channel activity going on somewhere, and that we're blocking it with tolbutamide. So what I'd like to think is that we've got some excess KTP channel activity going on somewhere in the conducting tissue. Why am I showing this? Oh, this is just no. This is to show that if we do this effect in Qrs 6.2 knockout animals, which have no KTP, we don't get any effect of tolbutamide on the heart rate. And in conscious animals, we see more complex phenotypes. So again, we can see missed beats. Here's one where you see a P wave, QRS wave, P, and a missed beat, P, QRS, P, missed, P. So this is 2-to-1 block. And occasionally we'll see this effect, which is a ventricular tachycardia, which is actually apparently quite rare in mice, and we never see anything like this in wild-type animals. And I didn't put the data up, but we see that the incidence of this kind of event gets more and more regular as the animals progress towards death. We have actually recorded telemetrically some of these animals all the way to death. So what we think is happening is that this is leading a progression towards a sudden death in the animal, and maybe what we're seeing is a fibrillation in a mouse. Now, I'd like to think that some of this is going on because of effects in the conducting tissue, and this just shows some slides from that were generated by Helena Dobrinsky, who is with Mark Boyett now at Manchester University. And what they've done is section mouse hearts. This shows crista terminalis, and this is the SA node region of the heart. And they've stained it here for connexin 43, and the lack of connexin 43 marks where the SA node is. And what you can see at the fluorescent level is that we have very high fluorescence of the transgene. This is from our transgenic animals expressing Qrs 6.2 mutants. You see high fluorescence through the crista terminalis and through the SA node, even though there's no connexin 43 staining in there. So this means that we do actually have the transgenes expressed at high density in the nodal tissue. And this is where I think there might be some more evidence for the relevance of all this to pathophysiology, because there is essentially the same phenotype in mice that are overexpressing Qr 2.1. And I told you at the beginning that the resting potential of the heart is, of course, going to be set by Qr 2.1, the IK1 conductance, and KTP if it's there. And this will be true in any cells in the heart. And what Anatoly Lopatin and his group found, Anatoly used to be in my lab, he's been in Michigan for several years, and he's generated animals which are overexpressing Qr 2.1 under control of the mice in heavy chain promoter. He sees exactly the same effect, which is, in this case is a 2-to-1 block. So here's a missed beat, here's a missed beat. He also sees evidence for atrial fibrillation, which is what these brief excursions are here. I didn't mention it, but Tom has also observed atrial fibrillation in anesthetized animals. And again, he can see occasional, on recording the RR interval, you can see this simple 2-to-1 block. And so what? Yes, so my point is that overactive IK1 under myosin heavy chain promoter can cause this 2-to-1 block, and these animals progress to a sudden death, I should say. And so does overactive KTP channels, and both of them are expected to increase background K conductances. And it turns out now that there is, if you have gain of function in Qr 2.1 at least, this can cause human short QT syndrome, which is expected to predispose to reentrant ventricular arrhythmias. And it turns out that this mutation that has recently been discovered by well, I'm not sure who did it, but Pepe Jalife is the senior author. This mutation, D172N, is a mutation in the pore of the channel which reduces inward rectification. And this particular mutation we've all been looking at for 15 years in KR 2.1 in recombinant expression turns out to be present in humans. And in reducing inward rectification, it means that you get a bigger outward conductance. So it basically is a gain of function in the channel. It leads to a shortened QT interval and a peak T wave, which predisposes to re-entrant arrhythmias. And here's an example of the Qrs Complexes from three unaffected individuals in this particular family. And here's the two affected individuals, a boy and his father. You see that the Qrs T interval is much shorter in both of them than it is in normals, and the T wave is very peaked and very big, which means a big disparity of repolarization in the ventricle in this case. Okay, so conclusions. KTP channels are expressed at similar high density in the pancreas and in the heart. In the pancreas, they do inhibit insulin secretion in low glucose, and failure to close them causes profound diabetes. But in the heart, they only opening an extreme metabolic blockade, as far as we can tell. Knockout blocks preconditioning and hypersensitizes to ischemic stress. With KTP overactivity, we're finally pulling it out we had to overexpress a very ATP insensitive Qrs 6.2 and a pancreatic SUR1 isoform. But this can cause complex remodeling and probably a predisposition to arrhythmias and to sudden death. And I'll stop there. Pictures of the people, and you can see if you can pick them out in the audience. Thanks.
When you're doing telemetry on your double transgenic mice, a couple of questions. Could you catch the terminal rhythm? Like, what was the final?
Well, so they seem to show. Tom will correct me because he's been staring at these records for months now. But the essence, as I glean it, is that there is increased incidence of tachycardia and not tachycardia, of ventricular tachycardia and fibrillation periods. But then eventually you see a loss of ventricular beats and a dying atrial beat.
Have you seen mice in general, or in these mice, like bundle branch blocks? Is that something you detect in the mice?
So what we see is a slight reduction of the PR interval. So that's the only thing we've detected. I'm not sure what else we can detect on
Widened QRS complex, aside from that wacky rhythm.
Yeah, did you see that, Tom?
No, not really.
I mean, I'm not sure what to make of it in the mouse because, you know, the final part of it is wrapped up in whatever the T wave is, so. I think what we need is sub-mapping, of course, which we talked about.
So if you look at mobile ATP concentrations in the heart by, say, P31 NMR, you find like levels of 8 or 10 millimolar.
Maybe not that high, but 5 to 8 maybe.
Okay, depending on the species.
Yep.
So I guess my question is what do you think the difference or reasons underlying the high density of channels in the heart are and the disparity of the amount of ATP that exists in the mobile form and that which the channel is responsive to?
So the total ATP concentration by NMR is 8 to 10 maybe, and the free ATP is probably in the 2, 3, 4 millimolar range, right? And so that's all well enough to give significant inhibition of the channel. With ADP present, at say 3 millimolar ATP, that'll put you onto the foot of the activation curve, so several percent in an excised pattern. Now, what's in the pancreas, you don't need to activate very many to completely suppress insulin secretion. So 5% activation is probably enough to completely suppress, to stabilize the rest of it. Yeah, to keep it hyperpolarized. In the heart, Well, what I said was 1% is enough to give 50% shortening, and my modeling with the Noble model, maybe 10% of maximum was enough to significantly suppress sodium current activation, significantly increase the stimulus that you need to get an action potential. And by then the action potential is so short that it's kind of just a spike anyway.
So why do you have so many channels?
Yeah, that's
and
the
such
question. I don't have a trident and a big long white beard, and that's the question. So I don't know why they're there. I mean, teleologically, I keep saying these are not there at a very high density for no reason. Plus, as I like to point out, this channel is a huge protein. That's even ignoring any other subsidiary proteins. Just the 4 SURs and the 4 Qrs 6.2s that make up the core complex of the channel contain 76 transmembrane helices. It's a megadalton protein. It's a huge thing. Energetically, it's very expensive to build this, no doubt. So why is it there in such high density? I think, I mean, it's a kind of naff answer, but the answer I can give is that it's a very, the regulation by nucleotides is very complex and subtle. And I think it's a crucial link between metabolism, the cell's metabolism, and the cell's excitability in all the tissues that it's in. And so I think that in the heart it does have a crucial role, a crucial protective role, and this is sort of being dragged out a little bit from the knockout animals when they kind of beat them to death. They die quicker. So Andrei Terzik has done a lot of these kind of experiments, exercising and really pretty harsh stress testing. So harsh adrenergic stimulation, things like this. Animals which have the KATP channel knocked out have no KATP in the heart. They don't tolerate exercise stress as well. They don't tolerate adrenergic stress as well. They tend to die quicker. So that the LD50 for running them to death is low. So they're doing a protective role, and in us, it could well be. So getting back to these, well, first of all, there are people who have no functional KTP channels. There are also people who have these overactive channels, and there's no reported cardiac phenotype of either of them. But in both cases, these people have very messed up pancreases, and I didn't discuss it, but the ones that have no KTP, instead of having diabetes, they have the opposite disease, which is hyperinsulinemia, which is, again, lethal and leads to, not lethal, but leads to brain damage if at an early age if they're not treated. So they're under pretty severe medical medication anyway. So if, I don't know whether these children might end up being susceptible to sudden death at some stage because of lack of or gain of KTP channel function. But if there was, it's probably missed because of the pancreatic complications.
So I guess one of your arguments would be that the subsarcolemmal ATP concentration may be substantially different than the mobile ATP concentration.
No, I don't argue that, actually. And no, I don't think it's necessary to invoke that. What is?
Well, you went from like the 8 millimolar, say, to the 3 millimolar level.
Yeah. You get a little bit of
shift.
shortening. You have to go to 1 millimolar.
And then you want to make more ADP, so like the NAK ATP.
That's in the presence of ADP. So when I did those simulations are done in the presence of trying to mimic what is the rest of the intracellular components. So it's in the presence of ADP. So so the prediction is that
it's the same.
But in the heart, even if you are completely metabolically inhibited, this is what 31P NMR studies will show you, that the heart is almost, the ATP doesn't drop substantially until just before it goes into Riga.
Right.
So the ATP is not changing. What's happening is ADP is rising slightly, and that's very hard to measure. But ATP is always around 5 millimolar until you're dead.
Do you think the subsarcolemmal ATP could be changing?
It could be, but not very much. There is one interesting result actually, which is you can actually block KATP channels with Wabane in excised membrane patches, and the way it's working is the Wabane is blocking the sodium pump, which is the major consumer of ATP at the membrane, And so you get a little rise of ATP at the membrane. So I'm not arguing that there isn't some standing gradients to the membrane from the bulk. But I don't think we need to invoke big differences to explain everything we've seen except for this failure to activate, which would, in order to do that, you have to keep the ATP higher, which...
Okay, any other questions?
Just a very naive question. So if you have, in the heart, you have SUR2 type, right? Which makes it, makes the channel less sensitive to ADP.
Yes, I mean, our data suggests that SUR2 is less sensitive to activation by ADP than SUR1 is.
Right. And then you have just a relatively constant amount of ATP there. So is there any chance for the ATP channel, your channel, in any case, to be activated?
Well, I would say that the way we'd expect it to be activated is by ADP.
Right.
But the fact is, we don't really see it being activated until, I mean, I didn't show you the experiments with metabolic inhibition. If we metabolically inhibit it, of course, it comes on. But what we were expecting to see was that the channels being turned on in these transgenics under normal conditions. So we're trying to, we can, of course, turn the channels on with metabolic inhibition. You get massive activation, you get accidental shortening, etc., etc.
I see. So and that condition, what is activating the ATP channel?
Well, under those conditions, it is whatever fall of ATP there is and
It's
the
a
rise
fall
of ADP. Yeah.
Okay, I mean, it's still a mystery why this channel exists in the heart and cardiac cells.
Yeah.
At the same density of sodium channels.
Yep.
We all know what sodium channels do, but nobody knows what these channels do. John, another question, quick.
Oh, yeah, I was just wondering, do you think APD is controlling your remodeling thing? Is that why you're getting...
Yeah, so what's controlling the remodeling? I don't know. I mean, maybe during development the channels are a little more influential. Or maybe, you know, everything is in feedback, but maybe the little bit of overactivity that is there might be causing some accidental shortening. It's causing a 15% reduction of cardiac rhythm, which of course would cause a 15% insufficiency, I suppose, overall. And so maybe that then triggers this stimulation of the calcium current to counter what would other give us feedback. But if you reduce the rate by 15% to have a normal output, you've got to increase the contractility by 15% during each beat. So I guess it's some feedback like that. I mean, it makes some teleologic sense that would happen if you had this background conductance. We haven't looked at Anatoly's mice yet to see whether their calcium current... Actually, I think didn't Anatoly have some very weak increase in calcium current?
He didn't see a change in calcium currents.
Okay.
Do we know about any differences in levels of expression with ventricular versus AV nodal cells?
No, is the short answer. Nobody has looked at KTP in nodal cells as far as I know. And still to do.
Do you see such profound phenotype?
Yeah. But it's not, we don't know how to isolate the nodal cells. So we're waiting for somebody to do it for us, basically. Or have Tom go somewhere and do it. But yeah, there's some simple, obvious questions to look at there.
Okay, well, Colin, thank you for a fantastic...
What's happened is that the ATP is changed quite much, but even during modest metabolic rates.